And our first speaker is sitting right here, Tom Rando. If you want to come to the stage.
Welcome, Tom Rando.
Is there a pointer?
Yes. Okay. Let's give him a round of applause.
Okay, great. Well, good morning, and again, thanks to the organizers for having me speak at ARDD2023. It's so great to be here. Apologies to everybody who was here yesterday, because you have to hear me again first thing in the morning. I will give an introduction that will be familiar, because it will still be on the general topic that I talked about yesterday. But yesterday I talked about kind of aging and stem cell biology from the point of view of factors in the blood and factors that are released during exercise and how that influences stem cells and tissue repair. Today what I'll do is I'll go inward to the cell and talk more about things that happen inside the cell that change in response to these aging factors or exercise factors. And we've studied this kind of from an epigenetic point of view for a long time now, but I'll talk about a recent project that's taken us to study actually heterochromatin and metabolism, bringing these kind of three areas together. Okay, is there a pointer or not? Anyway, so this is a review, a figure from a review that I co-wrote with Anne Brunet and Peggy Adele. Anne Brunet did the lion's share of the work on this review, but I'm showing it anyway. And this just kind of illustrates stem cells throughout the body that are important for tissue homeostasis and tissue repair. There are some tissues that are highly regenerative, and the tissue we study is one of those tissues that is highly regenerative, and that's skeletal muscle. And this is just illustrating kind of a time course of repair from an uninjured muscle through day 3 after a necrotic injury. You see a lot of inflammation and early proliferation, which you can't see from the stem cells. And then after day 7, 14, and 30, that tissue repairs, essentially recapitulating development. So you get the same kind of changes that occur during the normal development of muscle. And the cell that is responsible for that remarkable regenerative capacity is this cell, the muscle stem cell, also called the satellite cell. And it was actually named this by the person who discovered it, who's Alexander Morrow. Discovered this in 1961. He was doing electron microscopy of muscle, and he discovered this tiny little cell sitting adjacent to, oh good, just in time, thank you. Here we go. So here's this tiny little cell sitting adjacent to these giant muscle fibers, just a tiny bit of which is seen here. But this little cell is very closely opposed, sitting under the basal lamina of that myofiber. And so that's really the niche of this stem cell. And he noticed many things about the cell, which I won't talk about today, but they're related to the biology, like the fact there are very few mitochondria, there are very little cytoplasm, it's almost all nucleus. But what he did notice, and I'll come back to that, is that these nuclei are very densely heterochromatic. And so this is a more modern view of that cell, and this is an image from Pete Zamet's work. And too bad it doesn't show up too well, but here's a beautiful single myofiber that has been dissected out. Each of these are these nuclei within the myofiber. These are one of the true few syncytia in the body, so one cell, many, many nuclei. But here's this little tiny cell sitting, sort of orbiting it. That's a satellite cell called the satellite cell. And these cells are rare. They are rare in the tissue. They remain quiescent in weeks, months, years if there's no injury, and then they get called into action, and they can have this remarkable ability to repair. But what we know is that as we age, we get a kind of a decrease in our ability to repair tissues. So tissues repair more slowly, less effectively. Almost always there's an increase in kind of fibrosis that occurs in tissues, and that's certainly true in skeletal muscle. So here's an illustration again of muscle repair in either a young mouse or an old mouse. And what we're seeing here is uninjured. This is a mouse after a single injury a few weeks later, after a second injury a few weeks later, and you can see it regenerates very well. And there's really almost no change even after multiple injuries. So really excellent regeneration. Whereas in an old animal, even after a single injury, within a few weeks, you start to see some impairment of regeneration. And you see little areas of unrepaired muscle, which will end up being depositions of matrix, and ultimately we will call that fibrosis. If you're given a second injury, there's even a longer delay in the regeneration. You see kind of persistent inflammation. So there's a real remarkable decline in regenerative potential with time, and we and others have studied changes that occur in the stem cell that are responsible, at least in part, for this declining regenerative potential. So this is a slide I showed yesterday, but this is the only one you'll see from yesterday. But we got into this field by asking the question, is there anything we can do about it? Is there anything that we can do to enhance the regeneration of an old tissue to make it repair more like a young tissue? And through a whole series of experiments, led us to do these kind of parabiosis experiments, and in particular heterochronic parabiosis, asking what happens to an old tissue if it's exposed to the circulating environment of a young mouse, and vice versa, but I'll talk about the benefit to the old animal. And indeed, if a young, in these isochronic pairs, these control pairs, the young regenerates very well. In the old control pairs, you start to see in this injury kind of scarring. If the old mouse has been paired to a young mouse, you really see remarkable regeneration without that kind of scarring, almost as good as a young animal. So we use the term rejuvenation. We use it in quotes. We try and be careful. And the question is this rejuvenation? And it certainly is restoration of youthful properties in terms of tissue biology, in terms of cell function, in terms of molecular features. The cells acquire more youthful features. But is that really rejuvenation in the sense of kind of reversing the process of aging? And I think this is a question for the field. And we really don't have a molecular definition of cellular age or a definition of the aging process molecularly enough to say that, I think. But again, from phenotypic points of view and molecular points of view, cells and tissues appear to be rejuvenated by this kind of intervention as well as others. So we thought about this, and we think about this kind of taking a page from something that's very familiar, and that is IPS technology. So something familiar to everyone. You start with a differentiated cell, and through these genetic factors, these Yamanaka factors, you can induce these cells to have lost all their differentiation features and they're pluripotent. That's the nature of the technology, and then you can redifferentiate them all the way from these single pluripotent cells into cells that can actually make an entire organism, including the germline. So this is truly pluripotency driven back to kind of stage one of differentiation. But the other thing that happens is that you start with a cell, no matter what you do, with a cell that has some age, however you want to define it. And in the process of that de-differentiation, they also rejuvenate, because these cells not only have lost all their differentiation features, but they have lost all their features of aging. And they have, in the same way, gone back to stage one of differentiation. They go back to an age of 0. So they get the aging process gets reset in this process, and then again, they can age while they differentiate and make tissues. And again, this process can go over, on and on and on. And it's not unique to IPS. I mean, clearly, somatic cell nuclear transfer does the same thing. So Dolly was not the first mammal clone, but the first mammal cloned from an adult cell. So that cell was from an adult animal, I think it was the bladder or something, and that nucleus was converted into an animal that was fertile, and there were always some issues on how healthy Dolly was, but lots of animals have been cloned from adult cells, and they can give rise to progeny. So they, too, undergo this rejuvenation in the process of somatic cell nuclear transfer. And to a much more fundamental level, this happens, this is almost the nature of life, which is, in order to form a fertilized egg, you take 2 cells from an adult animal, adult humans, whatever, that have some age, you put them together, and you reset the clock. It has to go back to 0. If it were not for that, we would be passing on our age to our progeny. We would go to extinction just by passing on our age. So this is just so fundamental, we accept this as a fundamental process of life. So this kind of rejuvenation happens. The question is, how much do we understand it? And the important thing is that in all these cases, this rejuvenation is coupled to de-differentiation. That is, you not only lose the aging features, but you lose all the differentiation features. And our question, with regard to kind of the parabiosis studies, but other studies in the literature as well, is can you uncouple these? Can you devise a way to reprogram age without reprogramming differentiation? And that's our question, is that what we're doing with parabiosis? We're actually not making pluripotent cells, we're just making younger cells of the same kind. And this is a fanciful image from a review I wrote with Howard Chang many years ago now, in which we kind of modeled this process of aging and rejuvenation as if the genome is sort of a film through which light is passed through a lens, and that lens is the epigenome, to form an image, which is the phenotype. And that with age, you get degradation of the genome, you get degradation of the epigenome, and you get an altered phenotype. And that with these kinds of interventions, whether it's parabiotic or pharmacologic or genetic, you're really targeting the epigenome. So you're restoring a youthful property of the epigenome, even if not having any effect on what might be genomic instability and genomic degradation. So you can restore partially, and there's an issue of perdurance, and this is a thing we think about a lot, partially the restoration of a youthful phenotype. So we think of it this way, and so we were looking for ways to focus only on reprogramming the epigenome, but Juan Carlos Belmonte beat us to the punch on this one, and he published his paper in 2016 in which he used the Yamanaka factors that they could pulse in vivo and in vitro and basically showed evidence that they were restoring youthfulness to cells in vitro and vivo and tissues without de-differentiating them. That's sort of the holy grail that we were looking for. And I think there's a lot of evidence now that this kind of partial reprogramming has that possibility. And we were working in this area, and this is from a paper with our collaborator Vittorio Sebastiano. We were working at Stanford, and we were using, instead of the cDNAs integrated, we were using mRNA vectors to transiently reprogram cells using mRNA that would degrade. And when we did the same thing, so we'd introduce these into cells in vitro or in vivo, and we would see evidence that we were rejuvenating cells in this case. Okay, so if this is right, if this kind of concept is right, if rejuvenation occurs via epigenetic reprogramming, what are we reprogramming? So what is the signature? What are the features that we're actually targeting in stem cells, or any tissue for that matter, to rejuvenate them? And so we've been studying kind of epigenome of these muscle stem cells for some time. And this is one of the earlier papers we published. And one of the things we found early is that with age, using ChIP-seq, the cells acquire this repressive histone mark, H3K27 trimethylation, at transcription start sites across the genome. And what was interesting is a lot of the genes that acquired this mark are genes that are not expressed. So what that's doing is interesting, but many genes that were expressed acquired this repressive mark and showed a decline in their expression. And the one category of genes that I'll talk about today is interesting are the histone genes themselves. So they occur in clusters. Here's a cluster of histone genes, and just by looking at transcript levels, there's some level in young, and they go down with age in each case. And they acquired this K27 mark. And we saw this both at the transcript level, we saw this at the protein levels, even for the core histones. And when we did a TAC-seq, we saw that these cells acquired a large region of nucleosome-free DNA. So as the histone levels go down, there's loss of histones, there's loss of nucleosomes. And this wasn't a new idea, because others had seen reductions in histone expression associated with aging in other systems. So Jessica Tyler, I have a couple of her papers here, showed this in yeast, that with replicative aging, there's a decline in histone biosynthesis, and that in fact, if she maintained histone levels, she could delay or prevent replicative aging in histones. So, really looking at it potentially causally, not just association. Jan Karlseder was looking at aging of cells in vitro. As cells senesce in vitro, they would also show a decline in histone levels. So there was this idea that maybe these are sort of drivers of these aging phenotypes. So we went back and looked at this using the parabiotic technique, and indeed, as we saw with function, heterochronic parabiosis not only restored the youthful function to these cells, but it restored youthful levels of histone expression. Okay, so we were really thinking about kind of chromatin more broadly through histones and nucleosomes, and again, we'd come back to this idea that these muscle stem cells, one is shown here, are densely heterochromatic. And that's compared to, say, these are nuclei inside the muscle fiber, very, very active. They're very euchromatic. So we started to ask, are there changes actually at the level of heterochromatin in these cells over time? And indeed, that's exactly what we're seeing now. So using electron microscopy as a start, we see a decrease in heterochromatin levels in quiescent cells, these non-dividing cells, over time with age. And this fits with a theory of aging that I really had not been familiar with, but there's a theory of aging called the heterochromatin loss theory of aging. And the idea is that with age, and this has been shown in species from yeast to mammals, that there is in fact loss of heterochromatin in lots of cells with age. And basically there's an opening of the chromatin, and associated with that loss of heterochromatin are decreases in these key marks like H3K9 dietrimethylation, HB1, associated with this opening. And this opening leads to, basically renders the genome kind of susceptible. Again, that's another idea that's out there very well. This is a paper that calls heterochromatin the guardian of the genome, apologies to P53, I guess, but heterochromatin also, that if you lose heterochromatin potentially through an imbalance of methyltransferases and demethylases, you open up the chromatin and then you end up with all these kind of risks of genomic instability. Everything from chromosome missegregation, which we've shown in these cells, you get retrotransposon expression, you get abnormalities in DNA repair across similar regions in different chromosomes. So it really kind of all badness for the genome, leading to cancer, but also associated with aging. So we looked at this in these stem cells, and indeed, if we look at evidence of DNA damage, we're using gamma H2X foci, increases dramatically with age in these cells as they age chronologically. So they're not dividing, but they acquire these DNA lesions. And consistent with this idea of loss of heterochromatin, we see a decrease in these marks of heterochromatin, K9 di and trimethylation. So again, this is all kind of consistent, but we looked at the levels of expression of the methyltransferases and demethylases that should be basically determining levels of K9 di and trimethylation, and we didn't see any changes, any pattern that would make sense for that. So basically, the levels of expression seem to be quite constant throughout age. So it wasn't a change in transcript levels or protein levels. So we asked, is it really instead, is it a change in enzymatic activity? So the levels aren't changed, but the activities changed, and what could possibly account for that? So this is what led us to think about metabolism. So here's another figure from another review I wrote with Anne Brené on this connection between metabolism and epigenetics. And the connection, there are many connections, but one in particular is that these chromatin-modifying enzymes all use metabolites from different metabolic pathways as cofactors. This is shown in a little bit more detail here. So whether looking at histone acetylation or deacetylation, or DNA methylation or demethylation, they all use cofactors acetyl-CoA, NAD, S-adenosylmethionine, alpha-ketoglutarate, as cofactors in these reactions. And if changes in these cofactors, so changes in metabolic pathways, have the ability to kind of modify the epigenome by modifying the enzymes they control. So we just started by doing basic metabolomics and asking, what is the metabolic profile of these cells as they age? And one of the most dramatic changes we saw was in one of these pathways. So with age, this is using LC mass spec, we see a decrease in S-adenosylmethionine and its product S-adenosyl homocysteine. We confirmed that by ELISA. So again, this is a key cofactor in methyltransferase reactions. And so we started to ask, are these actually connected causally? Are changes in metabolism affecting epigenetic features? And this is work being done by a postdoc in the lab, Jianming Kang. And so she started doing mostly pharmacology so far, so using inhibitors in these pathways. So using an inhibitor of SAM synthesis, Jianming showed she could reduce SAM levels, and that's associated with an increase in DNA damage. So we can make things worse by blocking a pathway. That's not always so surprising. But you could make things better by restoring SAM levels in these cells, either in vitro and in vivo. So in vivo, doing supplementation with intraperitoneal injection of SAM, she could show increases in these heterochromatin marks, K9 di and trimethylation, and increases in HP1, and indeed increases in heterochromatin. So just by supplementing this metabolite, it was restoring some balance that was lost with age. And that restoration of heterochromatin was associated with, again, a reduction in the DNA damage by either gamma H2X staining or comet assay, and that could basically be reverted from, so it goes up with age and it could be reverted by supplementation with SAM. And even though this is a kind of more complicated or integrative assay, supplementation of SAM could enhance regeneration of old tissue to become more like young tissue. So accelerates the regeneration that we see with age, which declines. So from the phenotype that we first started with, just supplementation of a single metabolite has a detectable significant effect. So we're working on these and other pathways, but just want to finish off by talking about when you study aging and pathways, it seems like you're always kicking the can down the road because you find something that changes, and then what causes that and what causes this? Will we ever get to the beginning? I don't know. So we're kicking this can one more spot down the road and asks what happens in the cell that leads to decreases in SAM levels with age. And so this is a kind of a more broad view of SAM metabolism in the cell. It's involved in a lot of reactions, but the one that caught our eye was this synthesis of polyamines. So from putrescine to spermidine, because SAM is utilized, it's consumed in these enzymatic reactions of polyamine synthesis. And in our metabolomic study, we saw an increase in polyamines in these cells with age. So the hypothesis is that this polyamine biosynthesis pathway is revved up in these cells. It's consuming SAM and thereby leaving lower levels of SAM available for other reactions inside the cell. And so Jiangmin uses inhibitors of either SAM synthesis, or in this case, inhibitors of spermidine synthase to kind of tamp down that polyamine synthesis and ask what does that do for SAM levels and these other features that we've been studying. And indeed, when she inhibits polyamine synthesis, she can see an increase in SAM levels in the cell, and that can be blocked by an inhibitor of SAM synthesis itself. So we're increasing SAM levels, and basically we saw all the things that we saw with just SAM supplementation by inhibiting polyamine synthesis. So increases in H3K9 tri- and dimethylation, and an increase in heterochromatin. So it can, again, go through polyamine synthesis through increasing SAM levels to restore youthful heterochromatin levels in these cells. And we saw, again, a reduction in DNA damage, which could be reversed by blocking SAM synthesis. So, okay, so this is our working biochemical pathway model, is that in the cell with age, and we've seen that there's an upregulation of these polyamine synthetic enzymes. I didn't talk about that. This pathway is revved up, and that consumes SAM in the process of forming these polyamines, leaving less SAM and less S-adenosyl homocysteine available for these transmethylation reactions that are important for striking this balance of histone methylation and probably DNA methylation as well, but we're looking specifically at these markers of heterochromatin and leading to these changes in heterochromatin. So just to end, back to this magic cell that was found many years ago, this cell that sits there not dividing but is highly protected during aging through this heterochromatin. Is this kind of, in this notion of kicking the can down the road, have we reached the end of the road? Is this really the guardian of the genome, and that there are many ways you can disrupt heterochromatin, not only the way we're talking, but through different kinds of metabolic stresses in the cell, but that's kind of a key bottleneck through which protection of the cell occurs, especially in these non-dividing cells, and there's loss of chromatin over time that leads to kind of aging phenotypes. And so I think the other thing, it opens up the possibility of potential targets, biochemical targets, for maintaining and restoring heterochromatin, retaining and maintaining youthful stem cell function. So that's a project that's ongoing, unpublished in the lab. Just want to acknowledge the people. Jiangmin Kan is the postdoc who's done all this work. I've mentioned our collaborators, Anne Bernay, Vittorio Sebastiano at Stanford. Peggy Adele is another colleague with whom we've been studying stem cell aging with Anne for many, many years now. And of course, I want to acknowledge the funding sources that have gone into a lot of these studies that are looking at stem cell aging, stem cell quiescence, the things that we really spend time thinking about. So I'll stop there, and I'll be happy to take any questions at this time. Thank you.
Thank you so much. Thank you so much, Tom. We have time, I think, for one question. We're a bit...
You mentioned that you didn't see a difference at the levels of enzymes, methyltransferases. Have you seen if the distribution of the promoter of line 1, for example, has changed, and it is connected to misexpression of line 1? Have you seen more line 1 in this?
Yeah. I mean, so we've looked at just expression of transposons and they go up. So I can't say that. I mean, I suspect that this loss of heterochromatin is having all of these defects that have been reported with loss of heterochromatin. To the extent to which that explains phenotypes, we don't know. We haven't tried to block that the way we have tried to block these other pathways, but that's happening too, and that can't be good. So.
All right. Thank you so much,
Great.
Tom. That
Thank
was
you.
really an amazing talk. Thank you so much.
Thanks.